now give the floor to my co-host, Dr. Palier, for the Q&A portion. Dr. Palier.
Okay, thank you very much, Dr. Nina. We have question for yours truly. Dr. Palier, we know that we still lack a lot of data. By the way, the question is from Dr. Sumalde. We lack data regarding virus that causes COVID-19. Nonetheless, may we know your opinion if the virus would be able to contaminate crops through feces of COVID-19 patients? Reports mention that the virus has been found in the feces of some patients diagnosed with COVID-19. However, it is unclear whether the virus found in feces may be capable of causing COVID-19. There has not been any confirmed report of the virus spreading from feces to a person, but researchers do not know how much risk there is that the virus could be spread from the feces of an infected person to another person. However, they think that the risk is low based on data from previous outbreaks of diseases caused by SARS and MERS-CoV. But for parasites, virus has a different biology and dynamics, but for parasites, they can remain viable for 3 to 5 years in the soil and other environmental samples. But for virus, the viability is few days or few hours. Thank you. And then question for Myra from Dr. Raimundo. From Dr. Raimundo, congrats, Myra, on your presentation. It's a big jump from Resitonia to melanocytes. Do those herbs you discussed being used in suntans or whitening lotions? What is the mechanism of action?
Thank you, Mameka, for the question. Most of the formulations that you can find in drugstores or in the supermarket would, if they're whitening compound, they would inhibit tyrosinase activity or tyrosinase expression. So tyrosinase is the key enzyme for producing melanin. So that's why if it has a, for example, I think most of the whitening compounds in the Philippines are made up of this arbutin or their derivatives. But in other countries, it's already banned. And for our work on this, there is a venture company based in Italy who wanted to develop lotion that can be used to promote pigmentation, because tanning is very big in Europe. Whereas for us, we want to have lighter skin color. In Europe, they want to have darker skin color. And one of our work research topics that I presented is this work on tyrottannin. And in fact, a Japanese company known as called Nano Innovation Lab has this line of products, and their product is called Suna Shot. And they also have shampoo that promote hair color naturally. It's not a temporary hair color that you can add or you apply on your hair and it will fade away. It actually promotes natural hair pigmentation. So if we published work on the effect on melanogenesis of tyrottannin, but if you want to buy this product, it's called Suna Shot, and I'm not sure if you can buy it in the Philippines, but I'm sure they can make it happen. So yeah, that's it, I think.
Yeah, okay. Thank you, Myra. A question for Dr. Cuevas from Paolo Arejona. The TMI you used in Marinduque is the same TMI we are using with 3 strains of trichoderma. For those who want to be composting in their backyard, can we also recommend TMI to be used? If yes, what will be the rate to be used?
Thank you,
for the question. Um, this TMI can be used, but it's too expensive because it costs more. Remember, it is made up of 3 different strains and 2 species of trichoderma. The activator is cheaper, much cheaper. If you are going to use it for your backyard composting, we recommend that you use the activator because it's cheaper. The TMI is really much more effective for enhancing crop production, increasing yield, for efficient nutrient absorption, and improving root architecture.
Okay. So thank you, ma'am. For Dr. Servancia, is it possible for bees to develop resistance against pesticides?
Unfortunately, it hasn't happened yet. So many studies, so all pesticides really harm bees. So I hope one day they will, but nevertheless, it also poses hazard because they will contaminate the bee products which are used for human consumption.
Okay. Thank you, ma'am. Question for Dr. Rabahante from Dr. Sumalde from UPLB. How many times did you monitor for the presence of pollinators which you used in the study? Would it be better to have a composite data from several monitorings of the same study site?
Yes, I agree. We need to consider more than one visit with the site. Actually, our standard is to have three visits, at least 3 visits, but more will be better. But 3 is enough based on some standards with FAO.
Okay. Another interesting question from Dr. Ronilo Flores. For Dr. Rabahante, thank you very much for your presentation. What is the feasibility of the use of a mathematical model in predicting or tracking microbial or parasitic sources and destination in an ecosystem and the consequences that it poses? I would like to juxtapose the question in the light of Dr. Palier's presentation. I'm particularly interested in the possibility of developing an integrated scale or risk guide for the creation of smarter decisions and policies.
Yeah, we can use mathematical model in many areas in biology, in microbiology, or in parasitology. Actually, I'm working with Dr. Palier in one of her studies with schistosomiasis in Mindanao.
Yeah, okay. So thank you. We have question for Dr. Cuevas. If there are other sources of potassium which can be readily accessed in order to mitigate these tailings affected cropping areas?
The easiest source would be the chemical fertilizer. You have potassium chloride, but returning the rice straw to the field is the cheapest, and rice requires large amounts of potassium. The requirement is, I think, is even close or higher, a little higher than the requirement for, or close to the requirement for nitrogen. And potassium, the rice straw is a good source. So potassium chloride, chemical fertilizer, is the easiest source, but it's expensive.
Okay. A question also for Myra, Prof. Myra. Does the increase in melanin content of Tcqa in Viva experiments give a sense of validation to the cultural belief that coffee leads to a darker color, say pinaglihi sa kape? Is there an indication that Tcqa can be used topically as an alternative tanning and sunbathing?
The photo that I showed during my presentation, in that photo, the Tcqa was applied topically. So we did not give Tcqa as an orally. It was not orally administered. So like what we do with coffee. So we do not have any study wherein TCQA was fed, but we have published work on orally administered TCQA and its effect on not TCQA, pure TCQA, but rather purple sweet potato that's rich in TCQA, and it has an effect on memory and learning. But we did not check the effect on pigmentation, skin pigmentation. So what I can say is that, actually TCQA is not, is also present in coffee, but there will be higher concentration in what is referred to as green coffee. So I cannot say if the saying "pinaglihis sa kape" is true. So maybe we need further research to validate that. Thank you.
Okay, thank you so much. We have several interesting questions here, but some of our panelists have already answered them through typing. For questions which were not entertained, you can email to us and we will throw them to our respective panelists. In the interest of time, we can no longer entertain those interesting questions. We shall now proceed to the synthesis. Dr. Cadiz may.
Yes, thank you. There are lots of interesting questions, but perhaps they can also type, the panelists, the answer to the questions. So to give the synthesis of our parallel session is Dr. Romel Sulabo. Dr. Sulabo is a professor of monogastric nutrition and the director of the Institute of Animal Sciences, University of the Philippines, Los Baños. He received his MS and PhD in animal science at South Dakota State University and Kansas State University, respectively. He obtained further trainings in animal science as a postdoctoral research fellow at Kansas State University and University of Illinois at Urbana-Champaign. Dr. Sulabo?
Thank you very much, Dr. Cadiz, and a pleasant morning or day to all of the participants. So I have the enviable job of synthesizing 7 excellent talks this morning, and I hope I can give justice to these presentations. Unfortunately, I only have 5 minutes to the synthesis. So we first had 2 excellent guest lecturers. The first was from Dr. Jan Leach of Colorado State University, and she talked about the impacts of climate change on plant disease and the challenges for agriculture. And she started with talking about the impacts of extreme weather events on agriculture. And as she stated, the picture for agriculture is very bleak, as yields will be severely affected. And at the same time, there is also evidence of increased greenhouse gases as demonstrated in rice. Challenge for us in managing these challenges is that most of the historical data would focus on investigating plant responses to single stressors. But what we need to, I think, understand is that phytobiomes are a complex system, that we need to look at it as a systems approach rather than individual factors and embrace the complexity. Thankfully, today we have advances that sets the stage. He listed 3 major advances that's helping us solve these issues. The first would be advances in systems level approaches. Second would be microbiome discoveries and its role in improving plant productivity. And this actually strikes me as something that which is also very close to us in the animal industry, as we understand now the important role of the microbiome. And then finally, management strategies such as precision agriculture. And to demonstrate the main point of Dr. Leach, she talked about the dilemma that we have between heat tolerance genes and disease resistant genes in rice. Enhancing heat tolerance may increase susceptibility of plants to pathogens or vice versa. So I think the main point here is that The phytobiomes are impacted simultaneously by multiple stressors, and studying their outcomes may help us more effectively develop successful and sustainable crop production systems. Our second guest lecturer was Dr. Mila Evangelista from the USDA ARS National Center for Agriculture Utilization Research. And she talked about assessing novel protein sources for value-added uses and markets, particularly pennycress, camelina, coriander, and Pcqa. And I kind of related to Dr. Evangelista's talk because she worked on evaluating novel plant-based proteins. That is also similar to the work that we do here at the Institute of Animal Science, assessing alternative protein sources. And in fact, we've also done some work with camelina. And what Dr. Evangelista provided us today is actually a model for evaluation, looking at the composition of the materials, looking at the protein functional properties of these protein sources, and see what would be the potential applications. So looking at solubility, foaming properties, emulsification properties, water holding capacities, and stability to heat. And I think this is something that we could take a look at as we explore alternative protein sources that we can use also here in the Philippines, not just for the feed industry, but particularly in the food industry. Then we had 5 presentations in our oral sessions. The first was by Dr. Rachel Gay Pallier, and she talked about revisiting farming practices, implications of zoonotic parasites, and food safety. And I don't know about you, but I'm kind of got a little bit scared from what Dr. Pallier said. And she talked about the dynamics of parasite contamination and transmission in agricultural setting through assessment of soil, water, and farm produce. And a lot of the data that she mentioned was quite telling. Some of the things that she shared was the higher positive rate of parasites in organic produce versus conventional farm produce. She talked about the impact of processing, such as composting, in reducing parasites in animal manure. She listed a few things on possible contamination sources, such as raw or improperly composted manure, contaminated water, presence of animals in the farm, lack of field sanitation, and cross-contamination. And this kind of highlights the importance of a coordinated effort to bring all the resources for us to be able to control foodborne illnesses. The next talk was from Dr. Mayra Villarreal, and she talked about natural products that regulate melanogenesis by targeting MITF. And I think what this provides us is the richness of natural products that we can explore. And I think the Philippines is very rich in these natural products. One of the things that she mentioned that was striking to me was that half of the drugs that have been released since 1994 came from natural products. And I think this is something that we need to explore further. And specifically, she talked about natural products from Thyrelea hirsuta that has been shown to regulate MITF, tyrosinase, and other melanogenesis-associated enzymes, and its impact on melanogenesis. And she set the tone. There is a need for us to focus more on natural product research. And I think this is something that we in the Philippines can explore further. The next talk was from our Professor Emeritus, Dr. Virginia Cuevas, and she discussed a study that they did in Mogpog, Marinduque in 2018 and 2019, looking at the effect of rice straw composting and the use of Trichoderma microbial inoculant as an adaptive strategy to address problems related to climate change in lowland rice cropping. And their results prove that the technique of incorporating rice straw during land preparation with the use of TMI activator can increase yield even in carpor contaminated paddies as much as 150%. And this can help mitigate the negative impacts of erratic rainfall pattern associated with climate change. The next talk was again from one of our professor emeritus here at UPLP, Dr. Cleophas Cervantia, the queen bee. And she talked about pollinator conservation in agricultural landscape. And this is something that is becoming more and more important for us, and to realize that 75%, in fact, of food crops benefit from animal pollination. And she discussed a conservation strategy. The first was to use agricultural inputs. Second is using mango blooms as an example, where you do not apply pesticides at antithesis, where pollinators are most abundant. Do not overhunt wild colonies, and to develop a bee pasture, which not only beautifies the farm, but also creates diversity in the farm operation. And finally was the presentation of Dr. Jomer F. Rabahante, where they developed a long-term monitoring protocol for pollinators and pollination. And this clearly shows the not just the potential, but the clear benefit of using quantitative protocols in agriculture production. And the work that they did was to really evaluate if there is a decline of pollinators in Asia that I think we need to understand. And what they propose is to use the new index, which is the P2P index or ratio, which can help us determine pollinator-plant interaction. And so finally, my last comments here is the session is about transcending boundaries in agriculture. And I think what is highlighted from the talks that we had today is the importance of managing the complexity in agriculture. And this may really rely on improving our understanding of the multiple factors that affect productivity either singly or in combination. And in light of the global pandemic, this has to push us to be more interdisciplinary or transdisciplinary in approach. And we have to maintain our optimism in science. So thank you very much, and congratulations to all of our speakers.
Thank you so much, Dr. Sulabo. I would like to thank all who attended this parallel session, especially to our guests and session speakers, for sharing their knowledge and expertise. I also thank the UPLB Information Technology Center, especially Mr. John Eric Dao, who has been with us since the preparation of this parallel session. I would like to acknowledge the hard work of our PAASE President, Dr. Gisela Concepcion, and for giving me the opportunity to host this session. Thank you, Dr. Concepcion. And as what our theme says, agriculture transcending boundaries, I hope that we will be able to forge collaborations in the near future. With this, we now end our session. Thank you very much.
Thank you so much.
Thank you.
Thank you very much.
Yes.
Thank you.
Participation.
We also invite them for the other parallel sessions of our online Congress, the APAMS 2020. They can just go to the PAASE webpage, paase.org, for the remaining parallel sessions.
And in addition, there will be an evaluation form. The link will be sent to your respective emails within the week. Thank you.
Thank you to my co-host and to our rapporteur, Dr. Rachel Palier and Dr. Sulabo.
Yeah, thank you, Dr.
Okay.
Candice.
Thank you.
Bye now. Bye.